Hi everyone, in this video we're going to talk about amino acid reactions and how we can form proteins or polypeptides. So one important reaction that occurs between amino acids is oxidation of cysteine. So cysteine is actually the only amino acid that has this SH group. And remember, a long time ago, we talked about how if you have two SH groups on separate molecules, you can oxidize them so that they form a disulfide bridge. So this was a long time ago, so it's okay if you don't really remember this, but that means what we can do is if two cysteine amino acids are near each other, so for instance, here we have one cysteine amino acid with that SH group on its side chain. Here we have our second cysteine amino acid with that SH group. And if we add an oxidizing agent, we're going to get rid of those two hydrogens on the SH groups. Those are going to bond with oxygen to form water. And then the two sulfurs are free to form a bond called a disulfide bond. Now this is a really important reaction because let's say you have a chain of amino acids. Two cysteine amino acids in that chain could kind of wrap around to get closer to each other. So maybe, for instance, you've got a chain and maybe one of the cysteine amino acids is here, the other one is here. They could create a disulfide bond or bridge, and that's going to hold the chain of amino acids together in that spot. So this can really help form the shape of a protein or a polypeptide. Now, if we form a chain of amino acids, what we're really doing is forming peptide bonds. So peptide bonds are also called amide linkages. So down below we can see from a while back we talked about how carboxylic acids can react with amines to make an amide. And this is why organic chemistry was really important. We needed those building blocks of information to show how proteins form. So remember with this reaction, the OH from the carboxylic acid and then one of the hydrogens from the amine will team up to form water. And then that leaves us free to form a bond between carbon on the carboxylic acid and nitrogen on the amine to make our amide linkage. And this is going to be the same exact thing with amino acids. So for example, down below, we have the amino acid glycine, and remember that the alpha carbon in the middle is just bonded to another hydrogen as its side chain in glycine. So it's the only achiral amino acid. And then let's say our other amino acid is alanine, and its side chain is a methyl group. So if we oxidize, or not oxidize, but if we heat these up, they can form a peptide bond. So what happens is we do form that same water molecule that we saw before, and we're going to form a peptide linkage between glycine and its carbonyl group and alanine and its amino group. So what you could kind of think of this as is the oxygen from the carboxylic acid is going to take two of the hydrogens from the nitrogen on alanine and create a water molecule. That'll leave one hydrogen behind on the nitrogen on alanine. Now what this is called as well is a dipeptide. So these are compounds that are made of two amino acids, and they're joined together again by that peptide linkage. The order of the linkage is really important because two different dipeptides can be formed when you have two unique amino acids that are reacting with each other. So for example, we could have also seen the opposite dipeptide form where glycine was on the right and alanine was on the left. So this is going to create a different molecule or a different dipeptide. Now, the order again is important because certain proteins need specific orders of amino acids in order to properly function. If any amino acid is out of place or if they're out of order, that could cause, for instance, a mutation in the protein and it might not function properly. So it is really important that amino acid chains are in the correct order. So we do want to be mindful of that. Now, sometimes it doesn't matter. Sometimes if you replace a glycine with an alanine, it's not enough of a difference. But if you start to see multiple amino acids changing, or if the protein's really small, it could make a bigger difference. So let's do a practice problem. Let's draw the possible dipeptides that could form if the following amino acids are mixed. So I'm going to give you two amino acids. The first one is cysteine. So let's draw cysteine. So we've got the amino group on the left, our alpha carbon bonded to hydrogen, and then our carboxylic acid with a negative charge. And the side chain for cysteine is CH2SH. I think previously I said this was a polar side chain, but it's actually considered a non-polar side chain. I was incorrect before. Okay, and then serine is the other amino acid. So serine looks very similar. Got the amino group on the left, our alpha carbon bonded to hydrogen, then the carboxylic acid group with a negative charge, and the side chain is CH2OH. Now this is a polar side chain, so I guess sulfur isn't quite as polar as I thought. Okay, so see if you can draw the two possible dipeptides that can form from the mixture of cysteine and serine. So pause the video, and then when you're done, we'll go over it together. All right, so the two possible combinations, we could have cysteine go first, followed by serine, or serine could go first, followed by cysteine. And here I'm just using their three-letter abbreviations. So let's draw the first one. So we've got our amino group from cysteine. So we're basically going to draw the entire structure of cysteine, but we're going to leave out that last oxygen. And now we're going to draw a new bond to the nitrogen on serine. So now we've got NH, and then we're just going to follow along with the structure of serine above. Okay, so cysteine's on the left, serine's on the right. And again, remember, we always want that amino group on the left because that indicates we have an L amino acid, which is what's commonly found in proteins. All right, so now let's draw the other structure, although it looks like I might run out of room here. Let's see if I can fit it in. I'll just draw kind of a dividing line there. So serine's going first this time, so we're going to draw that, but we're going to leave out the oxygen on the very end, on the right. Okay, now we're going to form our peptide linkage to cysteine. And then we're just going to follow cysteine's structure. Oh, I just barely fit that in. Okay. All right. Now, there is also the possibility that you could form a cysteine-cysteine dipeptide or a serine-serine dipeptide as well, but I kind of left those out. So again, it's very important which order these bond in, and there are ways that you can get around this. For instance, you can use what are called protecting groups. So you could protect one end of cysteine, for example, and that will prevent cysteine from bonding on that side. So let's say we protected the amino group. So then only the carboxylic acid group could react. So that's one way to prevent these mixtures of dipeptides, and then you would only form one of the dipeptides. Okay, so these are things, scientists have to think about when they're trying to, for instance, synthesize a particular order of amino acids. Now, let's talk a little bit about peptides, because typically we do find longer chains of amino acids. So peptides are an amino acid polymer, so that means many. So you have many amino acids bonded together, but they're of a short chain length. So a polypeptide is an intermediate chain length polymer, so it has less than 50 amino acids. Now a protein, on the other hand, is a polymer with more than 50 amino acids. So once you kind of get beyond that 50 amino acids, then we consider that a protein. But if you have less than that, it's a polypeptide. Now, typically when we're talking about amino acids within a polypeptide chain or a protein, we call them amino acid residues. So you might hear the term residue, or you might read about amino acid residues. That just means it's part of a polypeptide chain. So again, we're going to make sure that when we draw our peptides, the N terminal, so the amino group, is on the left, and then the C terminal, so the carboxylic acid group, is on the right. So down below we have an example of 3 amino acids that are bonded together. So we've got alanine bonded to glycine. And then this last amino acid is called valine. And notice that on the very left side, we have the amino group from alanine. So we would call alanine the N-terminal residue because it's on the far left. And then we have the carboxylic acid group of valine on the far right. So we would call valine the C-terminal residue. So N for amino, C for carboxylic acid. So let's go over some important peptides. A couple of peptides you might have heard of before are vasopressin and oxytocin. So these are hormones that are released by the pituitary gland, and they each have 9 amino acids in them. So they're considered nonapeptides because they have 9 amino acids. Now, both of these peptides have disulfide bridges because they both contain 2 cysteine amino acids. So if we look at vasopressin down below on the left, we've got a cysteine here and a cysteine here. So they can kind of, the polypeptide can kind of twist around until those cysteine amino acids are lined up, and then we get that disulfide bridge between them. And that's going to kind of hold the shape of the peptide. And then oxytocin has the same thing. We've got 2 cysteine amino acids, so they can form that disulfide bridge. Now, these two hormones are really similar. They only differ in their third and their eighth amino acid. So notice the third amino acid is what's called phenylalanine in vasopressin. In oxytocin, it's isoleucine. The eighth amino acid in vasopressin is arginine, and the eighth in oxytocin is called leucine. Now, just those small differences make a huge change in the characteristics of these hormones. So vasopressin, which is an antidiuretic, that's a hard word, antidiuretic hormone, it decreases urine formation, whereas oxytocin causes uterine contractions. So just those small differences in the amino acids makes a huge difference. Now, another important peptide is called the adrenocorticotropic hormone, or ACTH. And this is also released by the pituitary gland. So ACTH contains 39 amino acids, so that's a lot, but it's still not long enough to be considered a protein. It doesn't have any disulfide bridges, and if we kind of look through all of those different three-letter labels, I don't see the label for cysteine, and that's why it doesn't have any disulfide bridges. And then ACTH regulates the production of steroids by the adrenal gland. So remember, steroids are a form of lipid. And we talked about other steroids like estrogen, testosterone, etc. And then the adrenal gland is actually a gland found on the top of your kidney. And so those are pretty important. Okay. Next time, we're going to start talking about proteins. So we talked about amino acids, which are the building blocks of proteins. And then we kind of talked about the middleman, the polypeptide. So that had less than 50 amino acids. And now we're going to talk about proteins, which have more than 50 amino acids in a chain. So I will see you then.